Good evening. It's a real privilege for me tonight to introduce our annual prize lecturer. The annual prize lecture of the Physiological Society is its premier lecture, and it's given by a distinguished scientist each year. And I have to say, in this particular case, I doubt that we could find a more distinguished scientist. I'm really delighted to have this opportunity to introduce Carla Shatz to the Society. She has a stellar career. Her education, if she doesn't mind me talking about that, was in some of the most prestigious institutions in North America. But also, she did an MPhil at University College London, which, of course, is something that I feel definitely needs recording. Since that education and her postgraduate training, again with some of the great stars of neuroscience in the 20th century, some of whom are still active even now, she has gone on to develop her own expertise across a broad area of neuroscience, such that her contributions have been recognized by innumerable awards. And she is, without doubt, one of the neuroscience stars in the world. In trying to talk about her, I really feel that the easiest thing that I could do, and probably the best, would be just to quote from the citation from the Royal Society, for which she was elected a foreign member this year, because I think it just puts it in a way that I can't. It says, Carol Shatz is distinguished for providing fundamental insights into brain development. Her research into spontaneous activity patterns that are used to tune and validate neural connections transformed our view of the role of neural activity in circuit formation. By investigating the interplay between genes and experience, most notably through her work on the activity-regulated MHCI genes, she has opened important new vistas in the field of developmental neurobiology. The fundamental work that she's done has been critical for the evolution of our understanding of the mechanisms that shape brain circuitry and contribute to the very essence of our being. I can't think of a more illustrious statement, and so I'm really delighted to introduce to the Society tonight Carla Shatz.
Well, Michael, thank you so much for that nice introduction. It's wonderful to be here. I see many faces, colleagues. I see Peter Kirkwood, who actually put up with me while I was learning physiology at University College London, and I'm so happy that he's here today. And I have to say I'm quite honored not only to be your lecturer today, but also to be in this building with Darwin and others here, their collections and so on. It's kind of really cool. And this debate that happened, I actually went to the Galapagos this December, which was another experience. I could tell you about that, actually, if you'd rather see my pictures. Anyhow, so I'm really very, very honored to be here. And as Michael described, I want to talk tonight about how some of our studies have helped us, at least me, to understand how it is that experience actually can change brain wiring. And I guess even if you don't like it, probably even after this evening, your brain will be different from what it was before, unless you're sleeping the whole time, which you could be. Although for me, it's 10 in the morning, so it's pretty good. So as we all know, obviously, our ability to perceive and to behave and to think depends upon the quality and the details of the neural circuits within our brain. And the thing that I've become most interested in is how is it that during development those circuits are constructed? How are they assembled? And then how are they maintained? And of course, the obvious point is that they're assembled and maintained through sets of specific genes whose expression is regulated at different times in development. And the first set of genes that are really important are those that essentially wire up and form the basic blueprint of connectivity during development. But then, of course, if we were all just wired up according to that fundamental blueprint, you know, the eye goes to the visual part of the brain, the ear goes to the auditory part of the brain, life would really be very boring. We'd all be the same. We'd all be walking around like automatons. And what's so marvelous about us is that, of course, experience then works its effect on this fundamental neural circuitry, working to tune it up and to change synaptic connections in a way that really, in the end, means that just as our fingerprint is unique to us, so too are our neural circuits unique to each of us. And it's this process whereby experience can alter gene expression that in turn then can tune circuits and lead to perception and action that I want to talk about tonight. And in particular, I want to talk about how one goes about trying to discover and understand the underlying genetic mechanisms through which activity has to change neural circuits. So what are those genes and how do you discover them? And as you can tell already, I'm going to talk about the visual system, the mammalian visual system, and I'm going to select really a very simple underlying organization of the visual system to focus on, and that is the fact that even at the very earliest stages of visual information processing, the inputs from the two eyes are strictly segregated from each other. So, for example, the output neurons of the retinas, the retinal ganglion cells, send their connections, obviously, to both sides of the brain. I'm only showing you the left side, where those connections make synaptic contacts with neurons in the LGN that reside in separate eye-specific layers. So the ganglion cells in the left eye send connections to left eye layers, ganglion cells in the right eye send connections to right eye layers, and those connections are strictly segregated in the adult, such that individual LGN neurons receive monosynaptic inputs only from one eye, not the other. And in turn, those LGN neurons send their connections up to layer 4 of primary visual cortex. And there, the connections again are segregated from each other, but this time not into eye-specific layers, but rather into an interdigitated pattern of right eye, left eye, right eye, left eye, right eye, left eye, LGN terminations that form the basis of the famous system of ocular dominance columns that were discovered originally by Hubel and Wiesel. So it's this segregated pattern I want to use as an example to try to understand how does it come about during development. And in fact, what I like to say here is that this pattern, you know, you could say, well, it's so beautiful and so precise, it must be hardwired. But the amazing thing is that it's not hardwired. And in fact, many sets of experiments, including those from my own lab, have shown that the initial pattern of input from, let's say, I'm going to focus now on the retina to the lateral geniculate nucleus, is not in the adult pattern of connectivity. So again, just to recapitulate, in the adult, ganglion cells in the right eye send their connections to right eye LGN layers. Ganglion cells in the left eye send their connections to left eye LGN layers. But initially, in development, ganglion cells in the right eye send their connections to end really throughout the different LGN layers. In fact, the layers aren't even present initially in development. Same for the right eye. So that in development, individual LGN neurons receive functional synaptic inputs from ganglion cells situated in both eyes. And actually, I'm very happy to say that many years ago now, Peter Kirkwood and I actually recorded from LGN neurons and stimulated the separate optic nerves in an ex vivo prep and showed that these LGN neurons do receive inputs from both eyes in development. But then later, individual LGN neurons only receive input from one eye. So during development, there's both a structural and a functional remodeling of synaptic connections. And I mean really structural too, because it's not just that synapses change, but for example, this synapse here from the left eye is literally eliminated, while this synapse here from the right eye is strengthened and stabilized. So there's both synapse elimination and synapse stabilization and strengthening that happen during development to give rise to the adult pattern of connectivity. Now, again, many years ago, many people, first in my lab actually, including some wonderful postdocs and graduate students who've now all grown up and they all have their own labs. I'm very proud of them. We discovered that neural activity is required for this remodeling process. So if you block neural activity during this period of development when this synapse remodeling goes on, then the adult pattern fails to emerge, and this immature pattern of connectivity remains, and if anything, is even more exaggerated. So individual neurons continue to receive input from both eyes, and there are all these side branches that remain in the ganglion cell axons. And incidentally, I should also say one other thing here, which is that it's not that only 2 ganglion cells in the right eye and 1 ganglion cell in the left eye send their connections to the LGN. In fact, there are about 1 million ganglion cells in the right eye, about 1 million ganglion cells in the left eye, about 2 million target neurons in the LGN. So this is, kind of a big problem to solve, but it is solved beautifully through activity. And when we blocked activity, then, as I said, the eye-specific layers fail to emerge. Now, there was a kind of mystery, though, and the mystery is this, that in all mammals, this process of formation of the eye-specific layers actually occurs before vision is possible. So, for instance, in us, in humans, this is happening in utero toward the end of the first trimester. In monkeys, it's happening in utero, as was shown beautifully by Pashko Rakic. In cats, it's happening in utero, and it's over pretty much by birth. In ferrets, it's happening after birth, but before eye opening. In mice, it's happening after birth, but before eye opening. And in all cases, it turns out, this activity-dependent remodeling is happening before the photoreceptors are functional and are connected, through the interneurons to the ganglion cells. So what kind of activity could be necessary for this formation of the eye-specific layers? Because I assure you it's not vision at these early times in development. And to answer that question, we realized that perhaps the retina early in development might be spontaneously active, and perhaps ganglion cells might be sitting there in the dark, generating action potentials and sending those action potentials to the LGN. And so I'm going to make a long story short now because it's many, many experiments, lots of which were very fun. But to make a long story short, what we discovered is that early in development, there are waves of activity that sweep across the retina in local domains. And those waves correlate highly the firing of neighboring ganglion cells. And that local correlated activity is then being sent to the LGN. And then through a process of synaptic plasticity, sort of cells that fire together, wire together, out of sync, lose your link, a pitiful abbreviation of what must be an extremely cool and complicated cellular mechanism that we still don't understand completely. There's this remodeling that's being driven by the spontaneous, highly correlated waves of activity. And I'll just show you, we've studied the waves in a variety of different ways, but one way is to use calcium imaging. And at this magnification, you're sort of looking at the whole retina. And when the retina turns black, these waves are then, ganglion cells become active, and about a pixel is about the size of an individual ganglion cell. So these waves are happening spontaneously. You can shine lights on these retinas until you're blue in the face, and you don't get a response from the ganglion cells because the photoreceptors are not mature yet. So it's this highly correlated spontaneous activity that seems to be needed for this process of segregation. And it's this activity that we blocked, and with blocking this activity, showed that it's required for the formation of the layers. Then naturally, after birth and after vision takes over, then vision itself provides the activity and the correlation that's required for continued subsequent refinement and development of connections in the LGN, and also, of course, in the cortex and beyond. So many years ago, again, we started to think, well, how is it that activity is ultimately being translated into these lasting structural changes at synapses? And is there a way to learn something more about the underlying molecules and mechanisms that are involved in translating activity into structure? And so we reasoned that maybe activity, this spontaneous activity generated by the ganglion cells, is able to drive and alter the expression of sets of genes that are themselves required for the activity-dependent remodeling process. So what we decided to do is simply block activity, thereby blocking the remodeling, and then compare gene expression in activity blocked versus active LGNs. So we used tetrodotoxin, which blocks voltage-sensitive sodium channels and is needed to relay the activity from the retina to the LGN. We blocked activity with tetrodotoxin during the 10-day or so period when this eye-specific layers are emerging during development. Then two postdocs in the lab, Rod Corriveau and Jean Ho, dissected the LGNs in the two conditions, plus and minus activity, extracted the RNA, and actually in those days, it was before there were really good microarrays, which actually turns out to be lucky for us. I'll tell you why in a minute. So we used a PCR-based differential display to detect changes in gene expression in activity blocked versus activity present. And we found a number of candidates, one of which, when we validated the candidate by doing in situ hybridization to look for where the gene was expressed, we were very happy to see that it was highly expressed in the LGN and very much downregulated when activity is blocked with TTX, also expressed in other places. But this was good. So when we sequenced this particular candidate gene, we got a huge shock. And the shock is that we found ourselves in the wrong system. And I don't mean like we were in the auditory system or the somatosensory system. We found ourselves in the immune system because the gene that we had discovered that was downregulated by blocking activity was identical to a known MHC class 1 molecule. Now, I always thought that the nervous system was complicated enough. And the good thing is that I didn't have to learn anything about the immune system, which in my opinion is more complicated than the nervous system, because it has all the same crazy names for cell types, but they move. So they don't stay in one place. But in fact, the reality is that now we're on a very fast learning curve and still learning about the immune system because the candidate that we had discovered, as I said, is identical to this, was one of these MHC class 1 molecules. Now, as many of you probably know better than I, these MHC class 1 molecules are transmembrane molecules, here's one in yellow, that in collaboration with a light chain, which is the same, and is needed for cell surface expression, stable expression of most, if not all, MHC class 1 molecules. There's some exceptions. These MHC molecules present peptide to the famous T cell receptors. So in the immune system, this is the adaptive immune system. So this is the system whereby T cells circulating in the body actually perform immune surveillance and make sure that cells are either healthy or they're infected by viruses. Cells that are healthy are actually expressing on their surface, they're presenting self-peptides that have been produced by degradation, by protein degradation in the proteasome. But if the cells are infected, then they actually express foreign peptides from viral proteins. And T cells notice that. And through a process of signaling and release of cytokines, actually function to destroy the infected cells. Now, this was cool, but our problem is that we had actually stumbled on a dogma, the dogma being that the brain is immune privileged. And what had been thought to be the case, among other aspects of immune privilege, is that healthy neurons do not express MHC class 1 RNA or protein. It is well known that when the brain is damaged, neurons upregulate expression of MHC class 1 genes. And actually this upregulation is thought to occur through release of cytokines, often either by invading cells of the immune system or even by the local microglia. So that's cool, but what I'm going to tell you now is that not only is MHC class 1 present in the brain and neurons and upregulated during inflammation and damage, but also MHC class 1 is expressed at low levels, normally in the healthy brain, is located at synapses and is required for certain aspects of activity-dependent synaptic plasticity. Moreover, I'm going to try to convince you that there is at least one, and incidentally probably now a variety, of neuronal receptors in the brain for MHC class 1 proteins. These are not the T cell receptor, but rather they're members of the innate immune system. And that what we may be studying is really a much more primordial or earlier function of these MHC class 1 proteins, which are probably co-evolved both in the immune and in the nervous systems. Now, just to motivate you, or at least to motivate me, when we first made this discovery, I immediately went to PubMed and I typed in MHC class 1 brain, MHC class 1 autism, MHC class 1 schizophrenia, MHC class 1 Parkinson's. What were we studying? Were we studying anything that was known to have some kind of genetic link to some kind of brain disorder? And for quite a long time, there was, I would type in these, and there would be a lot of debates. So I would type in the most recent MHC class 1 schizophrenia paper would say, there's this really fascinating link. And then about 3 months later, there'd be a new paper, no link, and so on. But actually, in 2009, 3 papers were published back-to-back in the summer of 2009 in Nature. Here's one of them. All 3 of them, genome-wide association studies, all 3 showing that the most highly associated SNP was located at the MHC class 1, the HLA locus. Now, we still don't really know what that means, actually, although I'm told now that some of these are becoming even more significant. But I think what's interesting is, so I read these papers avidly and was very disappointed to see that no one referred to any of our papers at all. In fact, the authors wrote that this was really interesting because there were these vague and interesting connections, epidemiological connections, with outbreaks of flu and with inflammation and other things that would affect the immune system. And what I'd like to try to convince you of is maybe the function that we think MHC class 1 has in neurons is another way to think about how, first of all, changes in gene expression in these molecules could lead to some of the diseases such as, and disorders such as schizophrenia, but also another way of thinking about how inflammation and changes in the environment could also alter the way the brain is using MHC class 1. So just something to think about as a way of motivating my talk, certainly a way that motivated us. Okay, so how are we going to study these genes? This is a very large gene family, not only in humans, but also in mice. There are over 50 class 1 genes, and only a few of them, the top few actually, have been studied well. In fact, K and D and L, which are very well studied in the immune system and are known to present peptide to T cell receptors. So one of the first things we did was simply to do a lot of in situ hybridization and look as well as we could for the expression of MHC class 1 mRNA, and just kind of abbreviate a number of experiments that we've done. And I should say that we're not really great, molecular biologists. So I'm sure many of you could make better, more specific riboprobes. But so far, we think that in the cerebellum, for instance, the Purkinje cells express one of these classical MHC class 1 molecules, D. It also expresses K. They also express K. You can see the granule cells express mRNA for a non-classical MHC class 1 called T22. It's a non-classical MHC class 1. Throughout the rest of the brain, for instance, in the cerebral cortex, the deep cortical layers, neurons express another one of these non-classical MHC class 1s called QA1. T22 is expressed in the hippocampus. It's also expressed, for instance, here in the substantia nigra. D, which is this classical one, is expressed in cortex, hippocampus, thalamus, and so on. In fact, aficionados will notice this is barrel cortex. And D and K are actually highly expressed in sensory areas of cortex, including somatosensory and visual and so on. And incidentally, this is a picture from an adult mouse brain. And so these molecules are not only present during development, they're also very highly regulated. And some of them come up during development, like those in the hippocampus, while others, like those in parts of the thalamus, are present early in development and are turned down by adulthood. So there's tremendous dynamic regulation of just these few. And I mean, it's sort of amazing. And we don't really understand much about their regulation at all. So far, I've told you then that MHC class 1 genes are expressed in the LGN, the cortex, the cerebellum, hippocampus. We discovered them because they're regulated by activity during early development in the LGN. Actually, they're also regulated by seizure. They go up by seizure and so on. So it's kind of a smoking gun, right? So now we could ask, okay, we found this gene family by this differential screen, but are they actually, we really didn't find it. It found us because we were not looking for it. So we could ask, is MHC class 1 needed for activity-dependent synapse remodeling? So how would you answer that question? So actually, I asked my mom, I said, Mom, how would you prove that MHC class 1 is needed for activity-dependent synapse elimination? And immediately my mom said, I'd knock it out. And I said, that's very good, Mom, but which one? So remember, I told you they're all over the place. So now I'm going to make another really long story short by telling you that we think that at least two of them, we don't know actually which one at this point, but at least two, either D or K on a black 6 background, because remember, your MHCs are what make your strain of mouse, so L is not expressed in black 6 and so on. This is very complicated, so you have to know which strain you're studying to know which MHCs you will find there. But in black 6 mice, we think that D and K may contribute to activity-dependent remodeling through a lot of hard work just looking for expression, where are they expressed and so on during development. And it just so happened that our colleague, when I was then at Harvard, Hidepleu, had made a K and D double knockout mouse. So we asked him for the mouse, and he said, well, that's great. Of course you can have the mouse, but the mice are fine. They have brains, which, to most developmental biologists, if they have a brain, it's not interesting, because if you knock out the genes you're interested in, they shouldn't have a brain. But we were really excited, and the reason is we thought, well, first they have a brain, we can study them. But more seriously, when we block neural activity during these later phases of development and prevent the formation of eye-specific layers, for example, the mice continue to develop. They just don't segregate their inputs from the two eyes. So to all intents and purposes, if you were a neuropathologist looking at the brain of a mouse that had undergone activity blockade for even 2 weeks, and you cut sections through that brain, the mouse would have the brain and would have all the cortical layers, cell migration, lots of things happen in the absence of activity. So the brain is fine, outwardly, but the details of the connections are what are different in those brains. So we thought, this is good, we'll look at these mice. So what should we look for in these mice? Well, I think I just told you the answer. The way we found MHC class 1 is that we found that it was high normally and downregulated when we blocked activity. So maybe the mice that lack MHC class 1 have unsegregated, more immature patterns of retinogeniculate connections than normal wild-type mice. So the way we looked at that was simply to inject the right eye with a green tracer and label all the connections from the right eye with a green anterograde tracer and the left eye with a red tracer. And if you do that experiment in an older, after segregation has happened, wild-type mouse, you get a very nice segregated pattern. So here we're just injecting the right eye with green and the left eye with red. We're looking at the right side of the brain, the LGN on the right side. So the red eye, the right eye is injected with green. So there's a little ipsilateral layer sandwiched by layers from the left eye. That's exactly what you would see. And there's very little overlap or intermixing of connections in these segregated mice. And so the yellow pixels represent the remaining areas where there's actually signal from both the right eye and the left eye. So a little bit of overlap is left. When we did the same experiment in the KD knockout mice, what we discovered is that there's extensive intermixing of the inputs from the two eyes that remains even well after segregation. So this is at postnatal day 34. Segregation is supposed to be over at about postnatal day 14. And in fact, this amount of intermixing is almost the same as that present in the immature brain before segregation starts. So we got really excited, because what it suggests is that somehow K and or D, we don't know which yet, maybe both, are needed for structural remodeling of presynaptic retinal ganglion cell inputs. So remember, We think K and D are in the postsynaptic cell. Keep that thought in mind. And we're looking here at a presynaptic phenotype. So something interesting might be going on even transsynaptically. Okay, so now I'm asking you, and you have to be probably an aficionado, if this result reminds you of anything. And what I mean by that is other mutant mice that fail to segregate their inputs during this activity-dependent remodeling period. And it was striking to us that just at about the same time we made our discovery, my colleague at Stanford, although then I was still at Harvard, published a beautiful paper. Beth Stevens was his postdoc, and she's actually now at Harvard. I guess it's balance of, I don't know, I went to Stanford, she came back to Harvard. Anyhow, so Beth in Ben Barris's lab, they had just discovered that mice lacking C1q also fail to segregate their retinal inputs in the LGN. And the phenotype is literally identical. Without knowing it, we both had used really the identical techniques to look at this anatomical pattern of segregation. So somehow, and I can't tell you how, C1q and MHC class 1 may be involved in some kind of a process that is required for the remodeling and essentially the elimination of synapses during development. And what I think is fascinating is that just recently, and again, there's been this debate over and over again about the relationship of the complement cascade in genetics of schizophrenia. And again, just a new paper just came out saying, oh, it's involved. This is another genome-wide association study. And who knows what the next paper will be, but I find it really fascinating. So now I want to kind of spend a few minutes just talking further about, how does this work? How does MHC class 1 work? Okay, well, the first thing is, where is the protein? So I've shown you where the RNA is, but where is the protein? Because we're talking here about activity-dependent synapse remodeling. So to start with, if we just use antibodies on sections of brain, it's it's expressed at the protein at very, very low levels, and we have to use intensification methods to see any staining at all. And it's kind of hard to see it in conventional cryostat sections. So far, in my opinion, the antibodies are not good enough to give very good EM resolution. That's what's needed. Kim McAllister published an EM paper earlier this year which shows that MHC class 1 is at the synapse, and I feel that we need better antibodies, frankly. But the antibodies that we do have, some of them work pretty well in dissociated cells in vitro. And remember, I told you that hippocampal neurons express high levels of MHC class 1, including K and D, and we have an antibody that's almost entirely specific for K and D. It does recognize a little band on KD knockout westerns. But the point is, if you take hippocampal neurons, as Alex Goddard did, and you put them in culture, and then you immunostain for MHC class 1, in this picture, MHC is red, and I'm sure the guys at the back can't see that, but they're little red puncta along the dendrites of hippocampal neurons in culture, and also the cell bodies express MHC class 1 protein. And if we stain for postsynaptic markers like PSD95 that stain the spines and synapses, the postsynaptic density, then we see a lot of co-localizations, suggesting that at least in the culture neurons that MHC class 1, a lot of it is actually associated with the synapse. But we really wanted to see where MHC is in the brain, in the real brain, not in the culture cells. And recently, a colleague of mine at Stanford invented a new method called array tomography. It's a very cool method, which essentially allows you to treat brain sections as if they were electron microscopy sections. But with a special plastic, you can actually stain the sections using antibodies that actually often don't work in EM. So you're staining at the light level, but you can cut thin sections of the brain. So you can cut 70 nanometer sections, and you can cut them serially. You can take each section, as this is one section here, 70 nanometer section, through our favorite part of the brain, the lateral geniculate nucleus, at postnatal day 7, exactly when this remodeling is starting. And you can stain it with your first favorite antibody, in this case MHC class 1, which in this particular picture is green. And immediately what you see is there are little green dots all over the place, which is exciting, suggesting that there's this very punctate staining in the LGN. But now here's what's really cool. So then you can take that same section and strip off the first antibody, and then you can stain with your second favorite antibody, which in this case is PSD95. And then some of you will see, at least in the front, that there's some puncta here that turn yellow. In other words, both green and red are co-localized. Both PSD95 and MHC class 1 are localized. So these puncta are likely to be synapses. Then you can strip off your second favorite antibody, and you can put on your third, which in our case is actually synapsin, a presynaptic marker. It stains the synaptic vesicles. And then you can even keep going up to 10 times before the sections all fall apart. And so what you do is you array, you do the staining with one antibody, then you take a picture, then you go to the next serial section. So you construct these huge tomographs, basically, so you have it in depth for up to 10 antibodies, and then you superimpose all the images. And when you do that kind of experiment with 3 of them anyhow, in one, this is one ultra-thin section here, you can see already there are cases where MHC is co-localized with PSD95 puncta postsynaptic, with presynaptic markers, and with all 3 markers. So you can do this with many other markers, and I'm going to now make a long story short again, and say that then when you actually do a process whereby you offset the images to check and see whether they're there by, the coincidence is there by chance or is highly correlated. If it's highly correlated, there's a fall off in the intersection of different pixels. If it's by chance, it's flat, the correlation is flat. So when you do that, you see that actually MHC class 1 is highly correlated with synaptic markers, which is good. A marker for GAD terminals, GABAergic terminals, is not correlated with a marker for glutamatergic terminals. Whew, that's good. And what's also fascinating is that the markers we use for C1q are also highly correlated with protein for MHC class 1. In other words, also highly correlated at synapses. And that's consistent with what Stevens and Barris found when they did array tomography for C1q and PSD95 and so on. Okay, so the results of all these experiments suggest that MHC class 1 is located at synapses. We favor the idea that it's more likely to be in the postsynaptic side because we see actually a lot of staining of dendrites, not just the postsynaptic and presynaptic densities, but also dendrites. So that's our bias. We could be wrong. Wouldn't be surprised if we're wrong. So far what I've told you is that neuronal K or D, we don't know which, are needed somehow for synapse elimination, at least in the LGN, and and the protein is located at synapses. So I want to ask again, how does MHC class 1 work? Let's think about it a little further. So maybe there's some neuronal receptors. So maybe MHC class 1 in neurons might be binding, possibly even across the synapse, to receptors, perhaps expressed, on the presynaptic nerve terminal in my wildest dreams, and that it's really transducing a signal. Now, MHC class 1 protein itself, they're not thought to signal themselves. So they're thought to signal by binding to receptors, which then signal. So what kind of receptors might we expect to see in neurons? Well, now we just thought, and now when I say we, I'm talking about Josh Seiken and Tadzia Grandpre, 2 postdocs who were in the lab. We just thought, well, let's just bite the bullet, and let's look at what other receptors in immune cells are known to bind MHC class 1 proteins. So we'll just look at, they're not supposed to be in the brain, but we're going to ignore that, and we're going to look for them in the brain. And the way we're going to do it is we're going to do just in situ hybridization screen, looking for some good candidates. And so actually we did that. And of course TCR, many people had looked at whether or not T-cell receptor is in the brain, whether it's expressed in neurons, very good labs like David Baltimore's lab. And everyone agrees that T-cell receptor is not expressed in neurons. The only time you've seen T-cell receptors in the brain is in T-cells, which come into the brain. Good, and that's what we found also, which is good because we were finding things other people were finding for a change. But some of these other receptor candidates do have high levels of mRNA expression in very interesting patterns in the brain, and some of them now actually have been reported to the protein is there, and they've been reported to be in neurons. We decided to look at this family of receptors called the LILR, or PEERS, the paired immunoglobulin-like receptors, and we looked at peer B. And I'll tell you why we looked at peer B. In the mouse, there's only one peer B, which is good. In humans, there are already a whole bunch of these LILRs. In fact, there are 5 at least. So we looked at peer B in part because not only was it known in the immune system by work from Takai to bind MHC class 1 protein, including K and D, and signal, but also it's a very lovely Ig superfamily transmembrane receptor, and it's called, the immunoglobulin-like receptor is called an inhibitory receptor because the intracellular domains of the receptor have these immunoreceptor tyrosine-based inhibitory motifs in the sense that when they're phosphorylated, then peer B recruits SHIP phosphatases, SHIP1 and 2, and this then engages downstream signaling that is known to oppose adhesion and MAP kinase cascades. And this is really cool because these are the favorite cascades that we know that in the nervous system are required for synaptic plasticity. So we thought that's very interesting because this is somehow putting some kind of a brake on these signaling cascades that are actually needed for synaptic plasticity. I'm going to come back to this theme in a minute. So we thought, okay, we'll look and see where this is expressed. And we found the mRNA expressed in neurons. And then we took cortical neurons and put them in culture. And we found that peer B is expressed in the growth cones of cortical neurons just behind the leading edge of the growth cone where you can find synaptic vesicles, synapsin staining. So it's really expressed on the palm of the growth cone in a subset of cortical neurons in culture. And if you stain the mature brain, you see that peer B is expressed in all kinds of really interesting places. Huge expression in the nose, in our favorite part of the brain, the visual cortex, in the hippocampus and the cerebellum, and other places in the brain. Actually, for Ray Guillory, the thalamic reticular nucleus. Quite an unbelievable, beautiful, actually terminal expression. I don't know why it's there. But the one kind of really annoying thing is peer B is not expressed in the LGN at this time in development. We couldn't see it either in the adult or in the developing brain because the first thing we decided to do was make a knockout and see what happens in mice lacking peer B. And of course, if I asked my mom, what would you do if you had a peer B knockout? And the first thing she said is, I would look at the retinogeniculate projection. And so then I said, oh, but Mom, the peer B doesn't seem to be expressed in the LGN, so I'm not sure what we should do. And so she said, well, okay, don't worry about it. Look in primary visual cortex, which also has a period of developmental plasticity, the famous critical period, during which time eye closure can alter the strength of the representation of the right eye versus the left eye, the closed eye versus the open eye. This is the famous experiments by David Hubel and Torsten Wiesel. So what they showed is that normally in the adult monkey brain, in this case, there's beautiful representation of input from the two eyes into ocular dominance columns or stripes as you look down on cortical layer 4. So if you inject one eye with a white tracer, in this case a radioactive label, then all the ganglion cells in that eye will transport that label to the LGN, to the layer in the LGN that gets its input from the injected eye. And then the LGN neurons will transport it one further step up to primary visual cortex, where the label will accumulate in the presynaptic nerve terminals representing the eye that was injected. So here you see, here's the right eye representation, the left eye, the right eye, the left eye. And what they showed many years ago is that during critical period of development, if one eye is closed, even briefly, even as short as a week or so, then the representation of the open eye expands to take over almost all of primary visual cortex, leaving only just these little piddly islands of black in a sea of white. So the open eye, so this is a use it or lose it, an activity-dependent process, really beautiful, and I think the first and the most remarkable demonstration of how experience can really alter brain circuits. And during developmental critical periods, in the sense that once these stripes are formed, if the eye is closed in the adult, then they're not susceptible to these major changes anymore in connectivity. They remain stable. So it's only during this early developmental critical period. So we thought, we'll do this experiment in the mouse. And the good news is the mouse does have a critical period. Many labs, including Mike Stryker, who really pioneered the use of the mouse as a model, sort of genetic model. So the mouse has a critical period, and you can close an eye. And in many studies, the open eye has been shown to take over, either functionally, physiologically, or even anatomically, the neurons in the cortex. The only sad thing is that on the scale here, which is this stripe here is about 0.5 millimeter wide, the whole mouse cortex can be fit into like 2 right eye stripes and 1 left eye stripe. It's pitiful. That's the whole mouse cortex. But still, just like the monkey, if you close one eye, there's an expansion of the open eye during the critical period. So we can use that. So now we can take these mice lacking peer B during their critical period, and we can close one eye, and we can ask what happens. But first, let me tell you what happens to normal wild-type mice. And actually, in this experiment, just to make sure that there was no vision through the closed eye, we removed one eye during the critical period. And we used a kind of lazy man's very convenient way of visualizing the input from the remaining eye, the open eye. And that was to use induction of an immediate early gene. So it's very simple, either CFOS or ARC. The mRNA for these genes comes up very rapidly with visual stimulation. So you can put the animal in the dark briefly, take it out into the light, and all the neurons that are getting visual input upregulate expression of mRNA. So then you can do in situ hybridization. And if you close one eye, you can look and see literally the functional connectivity of the cortical neurons receiving input from the eye that gets stimulation. And that's what you're looking at the top. This is just a normally reared mouse that was in the dark briefly. We took it out, and now we're looking at the different cortical layers. And there's a nice patch of input into layer 4. And this is now you're looking at the target cells that are getting functional input from activity through the open eyes. So you're actually looking at the mRNA that's been upregulated in this in situ hybridization. And mice have a critical period in the sense that if you close an eye or take one eye out during the critical period, the area that gets input from the open eye expands. So this is a nice example of ocular dominance plasticity. So we figured that take the peer B mice. I guess I could say I asked my mom again, okay, Mom, so what do you think would happen if we do the same ocular dominance plasticity in peer B mutant mice? And my mom said, well, I figure if peer B is activity-dependent regulated gene and it's needed for plasticity, then in mice lacking peer B, there wouldn't be any plasticity, which is what many mice have, genetically mutant mice have been found. So I would think that the result would be even after eye removal during the critical period, there would not be this wonderful expansion of the open eye, the remaining eye. And to our huge surprise, actually, we discovered that there was more expansion of the representation of the functional representation of the open eye in the visual cortex in knockout mice. So this is amazing. So there's actually more plasticity than normal. And this was really cool because remember I told you that we think that the way these peer signaling works is to oppose the kinase cascades and the adhesion cascades, such as integrin signaling, that are needed for plasticity. So if you remove the brake, maybe you get more plasticity. Hand waving, but that's what we're very interested in following up on. Okay, so let me summarize what I've said so far. So what I've said then, we can calculate essentially a plasticity index for wild-type mice and for knockout mice based on the width of the area of arc induction by the open eye. And what I've just told you is that the plasticity index is greater in peer B knockout mice. Okay, so let's think about this a minute then. So I've given you now 2 phenotypes in 2 different genotypes. I've talked about KD knockout mice in the retinogeniculate pathway, and I've talked about peer B knockout mice and ocular dominance plasticity being enhanced. So if our little model here that I've been invoking is correct, and that somehow MHC class 1 signaling through peer B is required to regulate the extent of plasticity in the brain and the extent of remodeling in the brain, then we would propose that mice lacking K and D in their cortex might also have enhanced ocular dominance plasticity. Let's go to the same place and look for phenotypes in that one place. And sure enough, when Akash Datwani did a set of experiments looking at KD knockout mice, he discovered that KD knockout mice, just like PIRB knockout mice, also have enhanced ocular dominance plasticity. So that's consistent with this model. Doesn't mean the model's right, but at least it's a good starting place. Okay, I'm wrapping up, but it'll take a little while. So now we can ask, how does PIRB work? How's PIRB working? So this is interesting. So we noticed that this enhanced plasticity that we saw in the visual cortex of PIRB knockout mice reminded us of a very important, I think, experiment that Mark Hübner and Tobias Bonhoeffer published a number of years ago, in which they showed that prior visual experience can result in enhanced plasticity in the adult. So remember I told you there's very little ocular dominance plasticity in adult visual cortex. And that's reflected by the fact that the plasticity index in wild-type adult visual cortex is very low. There's a little bit. You can get a teeny little shift in ocular dominance if you close one eye. You have to really wait a long time, incidentally, to get that shift. But you can get it. Whereas it's very high during the critical period. Well, what they discovered is they could actually get more plasticity in the adult cortex by closing the eye during the critical period and then opening it and then letting the mouse grow up and then reclosing that same eye in the adult. And what they found was a larger ocular dominance shift. And that kind of reminded us of our PIRB mice that have a larger ocular dominance shift normally. And what I didn't tell you, but I'm telling you now, is that we also know that in the adult PIRB knockout mice, there's more plasticity than in wild type. And in fact, it's rather similar to the amount of plasticity that's present in wild type mice after a prior period of eye closure. So this is really interesting, and it made us think that maybe PIRB signaling is somehow involved in this persistence of plasticity that can exist after experience. And if so, in PIRB knockout mice, we thought maybe this second eye closure would be occluded in the absence of PIRB. And Maya Jurisic in my lab, who's been collaborating with Mark and one of his students, has actually discovered that in the absence of PIRB, there's this elevated plasticity in the adult, but you can't get more from prior eye closure. In other words, you really need PIRB during development to get this extra plasticity in the adult. Now, they made another important discovery, which is that this experience of early eye closure actually leaves a lasting structural trace in the visual cortex. There is a real anatomical substrate that persists after eye closure in wild-type mice, and that substrate is dendritic spines. So it turns out that what they discovered is that if you look at the apical dendrites, in this case they looked at the apical dendrites of layer 5 pyramidal cells, and they counted the number of spines on those apical dendrites before and after eye closure. And they did a really cool experiment where they could actually count in the same animal, in fact. And normally, as you probably know, during development, there's this synapse elimination that's going on in the cortex. And the number of spines during postnatal life are decreasing normally in development. And that's true in the visual cortex. But what they discovered is if you close an eye during the critical period, then there's an increase in spine number, which remains actually into adulthood in normal wild-type mice. And this then, they think, is the substrate that can be re-engaged for enhanced plasticity in the adult. That's what they think. It's all correlation at this point. But it occurred to us then that maybe PIRB mice have this enhanced substrate all the time. And that's why when you close an eye, even in the adult, you get more plasticity. So maybe they have more spines. So Maya has been counting spines in PIRB knockout mice, again, the apical dendrites of layer 5 pyramidal neurons, and this is very new, hot off the press, and what she discovered is actually there are many, many more spines on the dendrites of layer 5 pyramidal neurons in PIRB mutant mice. So this now, I think, is very interesting because what we think then is these spines, instead of being eliminated during development, are stabilized. And this is very reminiscent to what I already told you about the failure of elimination of ganglion cell terminals in the LGN during development in the absence of peer B. So it seems like somehow these molecules are involved really in elimination of synapse or regression of synapses. Okay, so I'm not going to go, I'm going to go through this really fast because I'm looking at the time, and I don't know about you, but I think I really would like to have a beer. So, you know, you could ask, is this a good thing or bad thing to have this extra plasticity? And we're starting to look at this now. But to make a long story short, I'll just say that we already know, Mike McConnell, who was a postdoc in the lab, we know that the mice lacking K and D actually are amazing Olympians. They learn the rotorod task very rapidly, much more rapidly than wild type, to a higher criterion, and they remember it for longer, which is amazing. And at the same time, they also have a lower threshold LTD at climbing fiber to Purkinje cell synapses. So there's a change in synaptic plasticity rules and also an alteration in motor behavior that are correlated with each other. In addition, I'm going to just show you this movie, but we can look at visual acuity in the KD knockout mice by training them to discriminate between gratings that are very easy to see versus very high frequency gratings. And what we just, and once they learn, they just can go right to the platform that's correlated with the high frequency grating, and then you can make the other grating finer and finer and finer until their performance drops to chance. And what's interesting is that both wild type and knockout mice have similar performance normally. So it's not this extra plasticity is bad for the mice. They can still see at least to this pitiful level of testing them. There are much better ways of testing them now. Glenn Prusky has developed very beautiful methods for testing. We haven't used them, but what's interesting is when we remove one eye during the critical period and let the mice grow up only with one eye, then the remaining eye actually serves them better. And the mutant mice can still see and discriminate gratings of a certain frequency at a time when the wild-type mice ability to discriminate has fallen to chance. So this is not bad for them. They can actually maybe even use that extra plasticity to see a little bit better. There must be something wrong with these mice, frankly, but we just haven't, we don't have the sophistication right now to test what's going on. So now I really am summarizing. So what I've told you today is that we think that MHC class 1 proteins, at least K and D, there may be many others, we haven't studied them, acting probably postsynaptically, and that's just based on the fact that we see so much protein in the dendrites of neurons, pyramidal neurons in culture, cortical neurons in vivo. So it's not just at the synapse, but it's in the dendrites. Probably signaling through peer B, and we put peer B presynaptic in part because we see this beautiful presence of peer B in axonal growth cones in culture and also in fiber tracts in vivo. That somehow this signaling is opposing cascades that are required for synaptic plasticity. So when you knock out either MHC class 1 or peer B, you actually get more plasticity, simple-minded model. And it's really somehow maybe C1q, it's collaborating with C1q in some way. And not only that, but somehow it's conceivable that these molecules are regulating something about the cytoskeleton or spine stability or something, because in the absence of MHC class 1 or peer B, there are more spines and there are more presynaptic nerve terminals. And now I'm just going to assert that we think that this might be a transsynaptic signaling system for deadhesion. So when the system is present, the signaling is to destabilize synapses. When you knock out the system, then synapses remain stable, and there's a failure of synapse elimination during development. That's the idea. And we think it's transsynaptic because of the phenotypes. We knock out the protein in one cell, but we see the phenotype in the presynaptic cell. And that's assuming you believe this model. It doesn't have to be this way. But this is something that we could really work on and try to prove. And finally, I just want to now put this into perspective of what, sort of in a pitiful, my way of thinking about generally molecular mechanisms that regulate plasticity. So what I've told you today is that in addition to the famous signaling pathways that are known to regulate synaptic plasticity from beautiful work in the hippocampus, from beautiful work in cortex and other places in the brain, where gene knockouts or downregulation of signaling through these pathways actually damps down or eliminates plasticity, we think we've discovered components of what must be a negative regulatory pathway, whereby when you knock out or damp down this signaling pathway, you actually get more plasticity. And ours are not the only molecules that are thought to be involved now. There are a few others, a handful of others, including NOGO receptor, and links. I'm happy to talk about those another time later. But what this seems like then is that really they're both accelerators and brakes on these molecular signaling pathways that regulate the extent or the rate of change of plasticity during development. And what I think is fascinating about this is that at least the brake part of the system involves molecules that really share a common molecular language with the immune system. I mean, they're really, used by the immune system. Actually, both PIRB, which is in non-T cells, it's in other kinds of innate immune cells, and MHC class 1. And it may be that MHC class 1 is really a missing link that connects inflammation in the immune system with changes in the brain that lead to disorders in memory and learning and synaptic plasticity, not just during developmental critical periods, but maybe even in adulthood. And that's enough speculation, so I really want to thank everyone for their attention. And now I really feel bad that you can't see all the names of people who've contributed to work in the lab, including some really nice collaborations with Stephen Smith and the array tomography, intrinsic signal imaging with Mark Hübiner and his colleagues in Germany, a wonderful behavioral neuroscience core at Stanford that allowed us to study the behavior of these mice, and also a collaboration that we started with Mark Tessier-Levine when he was at Genentech on PIRB and axonal growth control. Thanks, everyone.